Okay. So, gonna go through anaerobic respiration. So I did a poll a couple of weeks ago for one of the biology lessons, and the two runners-up were anaerobic and aerobic. So I thought, well, I'll go through anaerobic respiration today and aerobic next week. Let me just check, can everybody here? Um, yeah, okay. Yeah, so let's get down to anaerobic respiration. Quite a small topic, so only going to be an hour. Bit of A-level biology. This comes up in your A2 for all exam boards. This is an A2 topic, but it's not too difficult, so we're going to go through it. And it's one of those topics that you need to know well, so we'll go through it. Okay, doesn't matter if you do an OCR, EdXL, or AQA, all the same. So the early Earth, no oxygen, or very negligible oxygen. There were oxygen atoms, of course, made during a supernova or whatever. So there were oxygen atoms on the early Earth, but there wasn't any atmospheric oxygen, free oxygen like O2 as we see it. So living organisms had to develop methods of making energy, of doing respiration without oxygen. So they used to use different methods. Now those methods were most likely different to the anaerobic respiration we're going to learn today. Those methods had a little bit of similarity with aerobic respiration and involved proton gradients and membranes. But the anaerobic respiration we're going to look at today, mostly in animal cells, has nothing to do with membranes. So let's get on and have a little look at it. So let's get into it. Boom. Right, first up, a little AQA question to get you warmed up. So 16.1 shows the structure of ATP, and we can't do respiration without mentioning ATP. It's the energy carrying molecule. And unlike when you talk about, oh my gosh, I've got fluff all over my hoodie. Oh, it's come out the dryer. Okay, unlike when you talk about respiration, and you're never allowed at GCSE to say respiration makes energy. We can say respiration makes ATP because it's a physical thing that gets made during respiration. So, good correction there, Matilde. It's not adenosine, it's adenine. So if you said adenosine, you wouldn't get the mark. This is actually adenine. So name the circle component in figure 16.1 is adenine. I know we call it adenosine triphosphate, but there we go. Right, what type of reaction converts ATP to ADP, so adenosine triphosphate, to diphosphate? Who knows the answer? What's the breaking reaction in all of biology? So you've got the same old reaction. It's the breaking reaction in biology. Yep. So we're going to hydrolyze. That's absolutely right. Well done. You are smashing out the answers. So good on you. Good on you. Boom. Right. Yeah, guys, don't forget. Do me a favor. Do me a favor. Hit the like.
If you push the button.
Would you like it?
Like.
Yeah, yeah. All right. Enough of the stupid videos. Let's get on with the actual lesson. Okay, so some situations where we might get anaerobic respiration. Looking at here, I've got an image. We've got some plants. Why would these? I don't know. Is that grass? Whatever it is, I don't know. I just took that image. Yeah. Why would these plants? Parts of them would be forced to do anaerobic respiration. Which parts of this plant would do anaerobic respiration? Well, check it out. It's obviously rained quite heavily. You can imagine in a flood it would be a lot worse than this, but the roots will be deprived of oxygen, and anaerobic respiration do form parts of the nitrogen cycle, but this is just giving you an example of where anaerobic respiration can happen, which is in the roots of plants when it rains. So all plants, when it rains, the water fills the soil, and what happens there is normally they do aerobic respiration using mitochondria. Remember, eukaryotes, plants and animals, eukaryotes and fungi, some of them, well, plants and animals have mitochondria in their cells, and that enables them to do aerobic respiration. So that would be the first choice of energy production for any complex cell, any eukaryotic cell, because it makes so much more energy than anaerobic respiration. But in an emergency, a plant could live maybe for a few days if the water was really waterlogged, by doing anaerobic respiration, its root cells could stay alive underground. And remember, all this water will fill up all the air spaces in the soil, going down, if it's a flood, going down like meters, so the roots will be deprived of oxygen. And when the water fills the soil, it's 10,000 times slower for oxygen to diffuse into the root, so it can't last long like that. Okay, no problems. That's all right, that's all right. No problem, Alex. No, that's all right, don't worry about it. I just do you know what, because I'm a tutor, obviously doing the lives is a bit of fun, but also it keeps me tuned in as to like what I got to do. I've got to stay on it. It's going to be big changes in 2027, so they're probably going to revamp some of the exams and stuff. So got to, this helps me keep on top of it. Okay, of course, anaerobic respiration happens, of course, in animals as well. So like when we do intense exercise, so we've got an example in plants when the roots are deprived of oxygen, but we've also got when you do intense exercise. And of course, I don't want to be sexist, so let's show a girl working out as well intensely. Now, you don't have to actually be training and lifting heavy weights to do anaerobic, and it doesn't mean that she's only doing anaerobic. Sort of a myth sometimes is that our bodies and our cells only do one or the other. No, they do both. Anybody knows if you play football, you're going to use the most efficient way to make energy, or if you play tennis or badminton or any sport, your body's going to use the most efficient way to make energy, which is aerobic. But there are times when the demand, for instance, lifting weights or kicking a ball or serving in tennis or throwing a punch in boxing, there's times when you need extra energy to get produce the maximum amount of energy. And in that case, our cells will dig in, or certain cells in certain muscles will dig in to use a bit of anaerobic respiration. Yeah, it won't keep you, if you try and hold your breath, you're obviously going to do aerobic with the oxygen that's in your bloodstream, but once that runs out, you'll go full anaerobic, and you're not going to last long just on anaerobic. It just doesn't produce enough energy. So where in the cell, guys, does anaerobic respiration happen? Okay, and does anyone know where aerobic respiration happens? Whereabouts in the cell? I'm just going to have a sip of coffee and just make sure all my cables are plugged in, because you never know. Yeah, this probably helps with A-level applied science biology. You're going to need to know aerobic and anaerobic respiration as well. Yeah, so aerobic occurs in the mitochondria and anaerobic in the cytoplasm. Now, once upon a time, one of our ancestors, if we went all the way back through evolution about 3 billion years, we've got an ancestor that didn't have any mitochondria. Well, there wasn't any oxygen on the earth, so mitochondria weren't a valuable thing to have. Once upon a time, mitochondria themselves were a form of either bacteria or archaea. They were a form of ancient single-celled creature, and our ancestor that was a single-celled creature engulfed a mitochondria and digested them, or whatever happened. Some of those eukaryotic cells that engulfed the mitochondria didn't digest them, and the theory is that the oxygen levels became really high on the earth, and oxygen was toxic, very toxic. It killed, or it was very dangerous, and organisms either had to evolve to survive oxygen or not. And one of the ways was, if you had mitochondria in you, they could use up the oxygen and so lower the oxygen levels inside the cell and keep it alive, because oxygen's such, if you do chemistry, oxygen, of course we say oxidation, it's named after oxygen, even though you don't have to be oxygen to do oxidation. It's making things lose electrons, stripping electrons off of things, and that was really damaging. So having mitochondria would have been a benefit, but we are looking at anaerobic, and it's in the cytoplasm. So anaerobic respiration happens in the cytoplasm. That's the clear jelly-like liquid within the cell that the organelles are suspended in. Remember, cells are three-dimensional structures, so we've got all these other organelles. You've got a centriole there, there's some mitochondria. They've still got the remnants of their ancient history as a bacteria. They still have circular DNA, not DNA in a nucleus. Although it only contains a few genes, they couldn't live outside of us now. Most of the instructions for the mitochondria are inside the nucleus, but there are a few instructions they have for making a few proteins and things inside here. But it's kind of like evidence that they were once a separate living thing. They have their own DNA, and there's various other bits of evidence for it as well. Okay, and then you've got the rough endoplasmic reticulum, ribosomes, what's that, the Golgi apparatus, something like that. Okay, anywho, vesicles, whatever. Let's get on. So let's look. Both do glycolysis in the cytoplasm. Yeah, anaerobic is in the cytoplasm. And do mitochondria need the DNA to make enzymes? I mean, technically the nucleus could do all that stuff for them, right? But what was the nucleus could make all that stuff for them. So there is a bit of interest there as to why they've kept a few genes. It makes us realize that why have they kept a few genes? I mean, they're no longer a separate living organism. They were at one time a symbiotic separate organism, we suppose. But yeah, so they've still got a few genes left. Maybe they make a couple of the enzymes that are really important. But I think the nucleus makes most of the stuff for the mitochondria now. Whether or not they will eventually lose all those genes in like another million years, if you came back and looked at a eukaryotic cell, maybe all of the DNA will be in the nucleus. But the chances are that the few genes they have left are really vital to be in the mitochondria for some reason, because otherwise natural selection would have said it's easier to not have them in there. So for whatever reason, I don't know the reason, but it must have been a good reason why they kept those genes, the few that they have. Okay, let's look at glycolysis. So anaerobic respiration comes in two parts. One of the parts is called glycolysis. The other part is called fermentation. So the anaerobic respiration that happens in animals and plants that we're going to look at, is broken into two parts, glycolysis and fermentation. Glycolysis also is part of aerobic respiration, which I'm going to do next week because it's a bit long. All good, Guy Patton? All good, all good, all good. So here you've got a glucose molecule, and you have to know the structure of that if you're doing A-level biology. Does anyone know if that's alpha or beta glucose? I'm just going to sip some coffee. There's a way you can tell if this is alpha or beta glucose. Yeah, it's alpha. Well done, guys. It's alpha. And beta glucose isn't really used in respiration. By humans, it's definitely not, because you can't eat grass. You can't break down cellulose. Cellulose, glycosidic 1 to 4 links. We don't have the enzymes that break that down. So we have enzymes that break down glycogen and starch, amylose and amylopectin, and we can turn those into alpha glucose, obviously, but we can't really access beta glucose. We just poo it out. So, or wee it out or whatever. We get rid of the beta glucose, so we're going to excrete that out of our body. Now, some herbivores like cows have certain bacteria in their stomachs. I know some of them have more than one stomach. I can't remember how many, but they have bacteria in them, and they can actually access beta glucose and use that for respiration. We don't. We use alpha. So yeah, hydrogen's above on carbon 1. That gives you a clue it's alpha. And the first thing that happens to glucose is it becomes glucose phosphate. Now there's various complicated names and steps in between here. You don't really need to know all of this. I'm just going to do a little bit of it, but you only really need to know the major details. So we're going to come on to pyruvate Vanchpuri. We're coming on to that in a second. So yeah, the hydroxyl is below the carbon, Mathilde, on alpha on carbon 1, or you can think of the hydrogen above on carbon 1, and the hydrogen would be below on carbon 1 if it was beta, and humans can't use that. Of course, what's been added, I put a little question mark there, what do we call this that's obviously been added to this glucose molecule for it to be called glucose phosphate? It's been phosphorylated, yeah, or partially phosphorylated at this stage, yes. It's been phosphorylated, and what would we need to use to add phosphate? So if you're going from glucose to glucose phosphate, what are you going to use to get that phosphate and add it? You can see the PO4, and it should have 3 minuses around it, but only 2 now because it's bonded there. So where are we going to get that? Fave mnemonic, alpha below, beta above for where the hydroxyl group is. Okay, yeah, ABBA. Oh yeah, ABBA, the 1970s pop band. Were they Swedish? I think they were Swedish. They were like the biggest European pop band for a while. Huge. Yeah, very good, Matilda. It's from ATP being hydrolyzed. So the hydrolysis of ATP provides the phosphate. And this has got a funky name. We actually say at this stage, the glucose is phosphorylated. So you have to know that. And we call this substrate level phosphorylation. Well, yeah, and it's because we're adding phosphate here to this substrate, this glucose, and we're going to make pyruvate, as someone mentioned there. Do you need to know these names like glucose 3 phosphate or whatever, or glucose phosphate or hexose bisphosphate? I don't think they ever really ask these names. So, but 280, the main thing you have to know is 2 ATP are hydrolyzed. So actually glycolysis uses energy at the start. So we're actually using energy in the process to make energy. So you've got to spend a little bit of energy to make net overall energy with glycolysis. So 2 ATP are used, and that gets us to this stage, hexose bisphosphate. The phosphate makes the glucose really unstable, so it's rapidly going to break down from this point. Yeah, no problems, Guy Patton. You will definitely need this later on. It does make it unstable. That's right, Matilda. It makes it unstable. And then we have 2 triose. The hexose bisphosphate doesn't last long because it's so unstable. The bonds snap and it becomes 2-TP, triose phosphate. I don't know if it's glycerate 3, whatever it is, but yeah, 2 triose phosphate. So 2 3-carbon molecules. So started off as a 6-carbon molecule, kind of became more unstable as it was phosphorylated by the use of ATP, and then it becomes 2 TPs. Okay, each TP has 1 phosphate group attached. Now what's going to happen? Well, what difference has happened? Nothing has happened as of yet. So it goes from hexose bisphosphate, and we've got 2 of these triose phosphates, and then it becomes 2 pyruvates. Have a look at the difference. What's the difference? The phosphate's gone. So where's the phosphate gone? So it goes from 2 triose phosphate to 2 pyruvate, and the phosphates are taken off. They're stripped off, and what happens is we actually 4 ATP are produced. So yeah, we get 4 ATP made. So overall, yeah, like that's a lot to remember, right? Oh my gosh, can't spell pyruvate. What's that? What's been added to make triose phosphate into pyruvate? Oh god. Yeah, be nice if I could spell pyruvate. Anywho, let's not worry about that. The main thing you got to know, first of all, glucose is phosphorylated, and then what's happened here? The glucose has ended up being oxidized. Okay, so electrons have been lost, and ADP has been turned into ATP. Now we used 2 and we made 4, so what's the net production of ATP? Yeah, Matilda's already said it, beating me to it, 2 ATP. Hi, Isha. Nice one. And yeah, you've got a couple of years before you do all this crazy stuff if you take biology. So glycolysis, we've also got to know 2 NAD is used, and they pick up 2 of the hydrogens from the triphosphate and they become 2 reduced NAD, and they're going to play a really important role in the second step of anaerobic respiration. So you might say this is too much for my brain to hold on to, too much information. So with any of these processes, rather than trying to learn all of them, so if you're in year 1 and you're like, oh, I don't have to learn this yet, just learn the summary. I'm going to go on to the summary of it, because all of these little details, most probably you'll never get asked. So the summary points, the key points are the main things. So the triose phosphate loses hydrogen ions, so it is oxidized to form pyruvate. So overall, we could say glucose is phosphorylated and oxidized to make pyruvate. And the NAD gains hydrogen, so is reduced. It also gains electrons. It doesn't just gain hydrogen ions, it gains hydrogen ions and electrons to become reduced. Obviously, reduction is gain. Remember oil rig from GCSE. Pyruvate's like a substrate molecule. It's going to be used in it goes off to be used in aerobic respiration in the mitochondria. Now, or if you're an organism that can do aerobic respiration, hopefully it will do that. If not, the pyruvate is used because really we're not interested in all of this stuff, the pyruvate. All we wanted to do was get this energy out from this series of reactions to get this net production of 2 ATP, and then we'll see how the pyruvate can help this go on. So, summary of glycolysis. this is what I would put on a revision card if I was summarizing glycolysis, which is the first part of anaerobic respiration and the hardest part. So glycolysis summary: glucose requires, it uses 2 ATP. We can say, if you want to know the extent of it, glucose is phosphorylated, then the glucose is oxidized to pyruvate. One glucose becomes 2 pyruvate. Didn't really put that in there, did I? So let's put 2 times pyruvate. This is very messy, so yeah, best to make your own notes on this business. And in that step, so in the first step, it uses 2 ATP when the glucose is phosphorylated. So you can know that the ATP was used to phosphorylate glucose. Then it became unstable and it broke down to form pyruvate. And in that process, it made 4 ATP. So we had 2, he says, make a little space, NATP, 2 ATP net is made. ATP is the small energy carrying molecule. And although we can't say energy is made, because energy doesn't, the law of conservation of energy, it only gets transferred, not made or destroyed. You do make ATP, which is the energy carrying molecule. Right, 2 reduced NAD is formed from NAD. Some people call this NAD plus. So if you look online and in textbooks, you might see that as NAD plus, but you don't have to call it that. People like to put a plus by it because it helps them. It's going to pick up an electron and then it won't be plus anymore. Okay, but at A level, we tend to just call it NAD or reduced NAD. Some examples call it NAD or NADH, so which is the reduced form. Okay. Now, what else? What else? This is quite a short topic, so it's only going to take about another 10 minutes of more waffle, and then we're going to do some questions on it. So, yeah, glycolysis. Here's a problem. This process, in order to produce these 4 ATP, we need NAD. Well, how much NAD is in a cell? Well, I can tell you there's only a few seconds worth in a cell. So if your cells want to stay alive, they better think of a way of getting NAD back. How could we get NAD back? We've turned it into reduced NAD in order to do this process here. It plays a role by accepting electrons. It plays a role in the formation of pyruvate and the formation of ATP. So we need to somehow recycle the reduced NAD to make more NAD. Otherwise our cell's going to die because it won't be able to continue glycolysis. And so that's where we come into the second step of respiration, and this is either anaerobic respiration. The second step is called fermentation. Now you've got 2 types: alcoholic in plants and yeast, and lactate in animals and some bacteria. Okay, some plants do lactate fermentation as well. Okay, let's whip through. So what you have here, pyruvate. Okay, if it's not going into the mitochondria, because if it's not going into the mitochondria to do aerobic respiration and it's just going to be used in anaerobic, in order to allow glycolysis to continue, this pyruvate is going to be turned into lactic acid called lactate, or you can call it lactic acid in your exams, and it's going to strip the electron and the hydrogen off of NAD when it becomes lactic acid, and that means this NAD is free to go back to the start of glycolysis and start and help with the oxidation of glucose and the formation of more ATP. So this is a vital step. You need to know that NADH or NAD must be regenerated from NADH in order for glycolysis to continue. Otherwise your cell's going to die. It won't last long because we'll run out of NAD in seconds to a few minutes. Okay, what next? You do need to know that we make lactic acid. Lactic acid is toxic. It's broken down in the liver and into carbon dioxide and water, the same things that we produce during aerobic respiration. And lactic acid it fatigues your muscles and makes them tired and sore. You know that from GCSE, hopefully. So yeah. And if you have a heart attack, it's the anaerobic respiration in the heart due to a lack of oxygen, due to reduced blood flow, that causes lactic acid formation as your heart cells try to do anaerobic respiration to produce enough ATP for muscle contraction, and you end up getting a real pain in your chest, and that's the symptoms of a heart attack. from anaerobic respiration. Now, in plants and some yeast, in some plants, not all of them, but in some plants, they do alcoholic fermentation. And this just means instead of making lactic acid, they make ethanol. Ethanol is an alcohol, and if you're a chemist, you know it's 2 carbons. So ethanol looks like this, and we're nearly finished with the content. It's literally, this is it. And then the content's done. So first of all, they go from pyruvate to something called ethanal. Ethanal is an aldehyde. If you're doing triple GCSE, then you're going to know this at triple GCSE. That's your ethanal. Ethanal looks like this. And then what happens? So first of all, a carbon dioxide comes off, and this is why I've got these bubbles over here. This is yeast bubbling away with some water or whatever, and these are yeast cells, by the way. It's a type of fungi, and essentially carbon dioxide's being released, hence the bubbles. 1 carbon has been removed. Pyruvate is a 3 carbon molecule, but it's gone down to 2. And then what happens is the reduced NAD is turned to NAD because the ethanol takes some more of the hydrogen, and the ethanol looks like this. So if you have a look, you can see ethanol has gained hydrogen there, on there, and where else? It has it's gained a hydrogen there. So ethanol's got 1, 2, 3, 4, 5, 6 hydrogens on the ethanol. Notice there was only 4 on the ethanal. So essentially it's taken the hydrogen off of the NAD and the electrons and turned the NADH, or NAD reduced, back into NAD, and now glycolysis can continue and can carry on. Okay, superbulous. Right, we're going to do some questions, so let's smash out some questions. I don't know if anyone's got any questions about this, but we're basically done on all of that's anaerobic respiration done. Nice and quick one for a Thursday evening. Next week I'm going to do aerobic and all the different processes in that, but I might do some AS biology as well. If people, I'll put up aerobic as one of the 4 options on a poll, and then you can choose if you want to do aerobic respiration or you might want to hit some of the AS topics. So I'll put up a selection, and if you want to do aerobic, I'll go through that next week, but I'll put it up on a poll. So let's do some questions. Yeah. So, all of my days. Has anyone got mocks at the moment or anything? Or has anyone got mocks? Anyone doing any mock exams? I don't know if people do mocks in January or November or... Okay, respiration is a vital process in living organisms. All organisms carry out glycolysis. The Krebs cycle also occurs in some organisms. The diagram shows some of the stages in glycolysis using hexose sugar glucose, using the hexose sugar glucose. This is, you've got French speaking tomorrow. Good luck, good luck, good luck. The diagram below shows some of the stages in glycolysis. So we've got glucose. It's being turned into phosphorylated glucose, the unstable version, and then it breaks down into 2, 3 carbon compounds. What is molecule R and S? Yeah, good luck with your French. Good luck to anyone who's got exams coming up in the next few weeks. I know quite a few people have got mocks. Yeah, R is ATP. Well done, well done, well done. And molecule S is probably PI or ADP, I guess. Yeah, ADP for S, because I guess the PI has joined to the glucose. So what's happened here is ATP, oh my days, moving my screen all around. ATP has gone to ADP plus PI. It's actually 2 times, so this would form 2 lots of that, right? But these bad boys have joined to the glucose. That's why they snapped off those phosphates to join to the glucose to make bisphosphate, hexose bisphosphate, which then quickly breaks down into like triose, and then it's turned into pyruvate. But so the answers to these are going to be ATP and ADP. This was an exam from the Edexcel Salters, Edexcel Route A exam board. So there you go. And some of the questions are AQA and some are OCR. All right, let's go. Explain why some ATP is broken down during glycolysis. So we've just kind of said that, haven't we? So what would be your answer to why is ATP used at the start of glycolysis? Why do we break down 2-ATP? What for? What is the ATP for? And there's a special term you want to say. This is a 2 marker. So I'm going to put the answer up. If you don't want to see the answer, you can pause the live, I guess, and have a go at this, see if you get both marks. But this would be the answer. So basically, any 2 of these, as it says. So ATP breaks down, it gives its phosphates to glucose. You could just say it phosphorylates glucose or donates phosphate to the glucose. ATP gives energy that helps to break down the glucose, as well as the phosphates. It uses the energy to snap the bonds. To produce 2,3 carbon compounds, you could name those. So you could say to produce glucose phosphate or to produce hexose bisphosphate. So you could actually name any of those names. I know I said you don't need to, but you can do to pick up marks. They'll always be on the accept kind of side. Yeah, to make it unstable, phosphorylate the glucose, provide energy to break bonds, and also to form hexose bisphosphate, or to form triose, 2 triose, or to form glucose phosphate. It's a whole load of names for the kind of intermediates in between. Now, this is a difficult one. This is an application kind of question because it's going to use names, and this is what happens in A-level biology. It's going to throw names at you that you've never heard of, like, what's phosphofructokinase? So phosphofructokinase is an enzyme that uses ATP to convert fructose 6-phosphate F6P into fructose 2,6-bisphosphate F26BP. The conversion of F6P by this enzyme is a rate determining step in glycolysis. This is shown in the diagram. So all of that is just to smash your brain, because that's what they want to do with biology, put you in an unfamiliar situation where you've got to use the knowledge that you've memorized. Okay, so you've just got to do the problem solving and spot what this is saying. Explain why ATP is needed for this reaction. Think about the previous reaction. What were our answers? That's all we got to do. Same thing. So you could pause and the video, the live, it's not really a video, it's a live, but you could press pause on it, and you could pause it and have a go at trying to get these 3 bullet points. I'm going to put the answers up. You want to put it in the context of this question, but it's the same thing we just talked about. So the idea is we want to break the ATP to provide energy to phosphorylate the glucose. Provides the phosphate group for phosphorylation of the glucose, or for F6P. So we need a phosphate to phosphorylate F6P. You can see it there actually. The P joins to the F6P and becomes, we can't say phosphorylate the glucose. This is why they've been a little bit cheeky here. They've stuck the phosphate onto there to make it 2 F26BP, so biphosphate. So the phosphate's being used to turn F6P into F26BP with extra phosphate. So you just got to kind of think on your feet. But yeah, hydrolysis of ATP provides energy. Yeah, that's right. It's needed for the energy. Yeah, ATP needed to phosphorylate fructose. Yeah. Good. Right. Okay. Couple more questions. Probably going to finish in about 10 minutes. Just going to do 10 minutes of A-level exam questions. So nice, easy one for you. Glycolysis occurs during germination. One of the products is adenosine triphosphate ATP. What's the other product of glycolysis? Or which of the others? There's actually 3 products to glycolysis. Which of the following is the other product in glycolysis? And Mathilde is all over this with the D. You're absolutely right. It's D. It's pyruvate. Of course, we're going to make NADH and pyruvate. Well done, Mathilde. Boom! Yeah, you had to have a boom for that. Okay, what we're going to do, don't get stressed out from the A-levels. That's what I'm here to help you work through, do some questions and that type of thing. So don't get too stressed. And you can always put up in the comments if you have a topic you want to go through. You can ask me questions about homework you got and that when I'm in this section of it, when I've gone through the knowledge. So
I can't handle the stress no more, dog. These damn A-levels.
okay, let's go. So, what other questions we got? Let's. Oh, that's disgusting. No one wants to see this question. Describe the different sources of the ATP that is used in muscle contraction during strenuous exercise. Okay, this was originally a 6 marker and you had to talk about aerobic, but you ain't done aerobic yet, so I'm skipping that bit. Really, I'm saying during muscle contraction, strenuous, lifting weights. Remember that lady who was lifting weights? What are the sources of the ATP that's used in muscle contraction? So it's a bit of a weird question, isn't it? What are they on about here? Describe the different sources of the ATP that is used in anaerobic respiration in muscle contraction. I don't like the wording to this question. You could pause and have a go. I'm going to put the answers up. So, all right, what they mean is, you get rid of that. That was one of the answers. I put a little title there, anaerobic respiration. This is one of the answers. Anaerobic respiration is the source during strenuous exercise. You probably could say aerobic as well, because it's not like you're just going to do anaerobic. You always do aerobic. And the reason is, like I didn't mention earlier, anaerobic only makes net 2 ATP for 1 glucose molecule, whereas the theoretical maximum yield of aerobic respiration, you can make 38. If in the ideal perfect situation you could make 38 molecules of ATP per glucose, it's often a lot less than that. It's often about 30 or 32. But in the ideal conditions, if you do so aerobic, having mitochondria being able to do aerobic respiration is a massive advantage to those organisms. In fact, you could say that without mitochondria there would be no multicellular life on the earth. 2 billion years ago, that's it, done. We'd all be single celled still. So without the mitochondria, we wouldn't be able to produce the level of ATP to clump cells together to make multicellular organisms. So yeah, it's vastly more efficient energy producing system. So anaerobic respiration is the source, and then we can talk about the substrates involved. So ATP is made from phosphorylation, glucose, net yield of 2, NAD. So it's just basically waffling on about anaerobic respiration. Didn't really like that question. Okay, what's this one? Yeah, we've done that. So yeah, same thing. I'm just going to put the answers up. So if you want, you could pause the video and have a go. Now, you're absolutely right, Mathilde, that aerobic is better, but there's organisms that just do anaerobic and they're like, no, it don't matter. I'm happy with anaerobic. You know, I'm occupying my niche and I'm good. So yeah. But yeah, it's definitely a much more efficient system. Right, I'm going to put the answers up to this. So if you were asked to describe glycolysis for 4 marks, you want to focus on the main bits. You don't need to get into all the dirty detail. So what happens to the glucose first of all? We use 2 ATP. We phosphorylate the glucose. Then the glucose becomes unstable and breaks down, and we oxidize the glucose, forming 4 ATP. And that oxidation uses 2 NAD that become NAD reduced, and it makes 2 pyruvate. So there's a net production of 2 ATP. So that's really the main bits you need to focus on. And this is a question from the OCR A-level biology syllabus. So they break this down, the glycolysis, into just the broad summary terms. You very rarely have to talk about things like hexose bisphosphate, if ever. So phosphorylation of glucose using ATP, oxidation of triose phosphate to pyruvate. Now the underlining of triose means they won't give you that mark in OCR. They won't give you that mark unless you say that it's the triose phosphate that's oxidized rather than the glucose. So overall, I've been saying glucose is oxidized, but of course the glucose doesn't exist after phosphorylation. It ends up becoming triose phosphate. So they're being a little bit specific here, but that's fine. You just have to know your exam board. So and there's a net gain of 2 ATP, and of course you make NAD reduced, which is vital for the fermentation bit where we recycle NAD. Okay. Okay, what would happen for viruses as they do not contain membrane-bound organelles? Would they only use anaerobic? No, Mathilde, viruses can't use either. Viruses can't use anaerobic or aerobic. They cannot respire because they're not alive. They have no cytoplasm. They don't contain the enzymes, proteins, and all the necessary things to make this work. Remember, in A-level biology, I mean, it looks pretty complicated, glycolysis, but it's actually each one it's even more complicated. If you were to really get into the biochemistry, you need an enzyme for each of these separate reactions. So enzymes control all these biological reactions. So if you went to university and you got deep on this, you'd be learning the name of the kinases and all the specific enzymes that are involved in these reactions. And viruses just don't have those. They've only got a protein coat and a little bit of DNA or RNA and maybe a couple of enzymes, or I don't even know if they have any, but they've only got a few proteins in there. Basically, they're very, very simple. So you might say, but then how do they stay alive? Well, they don't. They're dead. So how do they move? A virus moves when you sneeze. It's in your body fluid. It goes to someone else. The virus doesn't really exist outside of a host. This is a bit of a parasite, a virus. If it's on the table in front of you, it can't reproduce or do anything. Can't even move. It can't do anything. So yeah, get that sunlight in your bedroom and sterilize the viruses. Yeah, they can't get no, they don't respire at all, so they can't get ATP. They don't need any ATP because viruses is just a little bit of genetic code inside a protein coat. Viruses take over your DNA. They hijack your DNA. I mean, they don't hijack the whole thing. They just insert their tiny bit of code into your big long piece of code. So imagine like you've got a book with 1,000 pages. The virus just slips an extra page in between page 22 and 23, and it just has the instructions to say make more viruses, and your DNA goes and it builds all the proteins so the code is just for those basic proteins. Say there's 20 proteins in a virus or 50 proteins, so that code is just instructs when your ribosomes and all that, and transcription, translation, all that business, the mRNA, they just basically, your cell goes about its normal everyday business, but it starts assembling the proteins and the enzymes that are needed to make the virus. So it gives out the recipe for making more. And then the viruses burst out or damage the cell and leak out, and they go and infect other cells that they randomly bump into or come across and attach and inject themselves into. So kind of a pointless existence, isn't it? All they're doing is, but they are replicating, but they have to use living things to replicate. They are sneaky MFs, whatever that means, molybdenum fluoride, because I would never swear. But yeah, they're very, very sneaky, these viruses. And yeah, but remember, what makes a bad virus? Well, in evolutionary terms, a virus was why COVID was always going to become weaker and weaker and weaker. Look at the flu. The flu killed 20 million people in South America. If that's true or not, I don't know. But it killed half the population apparently when the Europeans turned up in South America. Look at viruses are pretty deadly, but if they're deadly, over time they're going to get less deadly because, of course, if a virus kills all the hosts, it's got no one to live in. So it's in the virus's best interest to end up like a cold. And again, when the Europeans went to Australia, millions of Aborigines of the indigenous population died from the viruses they brought there, the things like the cold. Now, the cold is not deadly now because everybody on the earth today are ancestors of people who survived the cold. So they have the genes and the immune system to beat the cold. But that's a good thing for the virus, because you'd say a cold is really successful, whereas Ebola is not very successful. Ebola's a crappy virus because it kills people, and then you don't have anywhere to live and you die. So pretty rubbish, isn't it? Yeah, it got less harmful, but it's the same with Ebola, isn't it? So if Ebola hits a village and there's 100 people living in the village, if it kills 70 of them, those people are now far apart and they can't spread Ebola between them, and then the virus dies out, because all those people die and the virus can't move or replicate or do anything outside of a living organism. So the virus dies off. So actually, if it didn't kill you quickly, it would be far more successful because it could then spread. And in fact, if it didn't kill you at all, really, and just then it could spread, and you could have millions and millions and millions of copies of the virus about. Anywho, let's zing on. I get waffling about viruses too much. So, which of the following statements A to D describes and explains the relative yield of ATP in anaerobic and aerobic respiration? So basically, which one makes more, etc. Kind of easy, this one. I think this is an OCR question. Anaerobic respiration produces less ATP per molecule of glucose because lactate is converted to pyruvate. Anaerobic respiration produces less ATP per molecule of glucose because NAD is not regenerated in oxidative phosphorylation. Anaerobic respiration produces more ATP per molecule of glucose because NAD is regenerated in oxidative phosphorylation. Anaerobic respiration produces more ATP per molecule of glucose because pyruvate is converted to lactate. Right, well, I think we can get rid of two of these, definitely. It doesn't produce more ATP per molecule, so I'm going to get rid of those. We've just said aerobic respiration, 38 molecules of ATP in the ideal situation per 1 glucose, and anaerobic, only 2 ATP per 1 glucose. So anaerobic is, do the maths, 38 divided by 2, 19 times crapper than aerobic respiration at releasing energy. So it's going to be A, yeah. We shouldn't be talking about oxidative phosphorylation. Oxidative phosphorylation is a more modern evolutionary type of phosphorylation. That is what happens at the end, in the final stages of aerobic respiration, where oxygen is the final, whatever it is, electron acceptor. And yeah, basically it's part of the aerobic respiration series of stages. So yeah, it's going to be anaerobic respiration produces less ATP per molecule of glucose because the lactate is converted to pyruvate. Okay. Whatever. I'm going with that. Oh, what the hell is this? This is just not even a question, so I don't know what this is. Oh, some of you will do anaerobic respiration practicals, and what they often do, they put coloured liquid in a tube. They do this more with aerobic, but you've got your glass tube, you've got your bung, you've got your yeast in here. Now yeast, like most things, can do, like plants and yeast can do aerobic or anaerobic. If you want to investigate anaerobic, you need to remove all oxygen from in here. Okay, you can't just put the bung in. We actually got alkaline, this pyrogalol, to absorb oxygen. So they put liquid in here that sucks all the oxygen from the atmosphere, and this ensures that the yeast are forced to do anaerobic respiration, and they're going to do alcoholic fermentation, therefore, and produce, obviously they're going to produce ethanol, and when they turn pyruvate into ethanal, they're going to release carbon dioxide. So we're going to get CO2 released. Now we can get a stopwatch and time how much CO2 is released per minute. And what's going to happen as the carbon dioxide is released, it's going to increase the number of particles inside this system. It's going to raise the pressure, and this coloured liquid is going to move in this direction. And what we can do is we can start a stopwatch. Check out the stopwatch. Incredible art. How do I do it? There's my stopwatch. 1 minute 26, whatever. And we can measure the distance that the red liquid, it's normally a red bead of liquid, that it moves. And it's going to be pushed by the pressure of the gas being released. So that's how they calculate the rate of respiration for this, by the distance the liquid moves. Yeah, you could just do a gas syringe and measure the volume of carbon dioxide given off a second, I suppose, but there we go. Right, let's do just one more question because it is time to finish up. Oh my gosh, what is this? So, yeah, we're just going to do this last question about anaerobic. Part of the body's response to fight or flight, this is when someone jumps on you and says, give me your money, and you're like, ah! Right, is to run away from the threat. Prolonged vigorous exercise put high demands on the body's metabolism. So we're like running away from, I don't know, somebody chasing us with a scary Halloween mask. The muscle cells require an adequate supply of oxygen for respiration. If insufficient oxygen is available, the cells must respire anaerobically. Figure 20.2 outlines the process of anaerobic respiration in your muscle cells. Identify compounds labeled D and E in 20.2. So we got glucose turning into something, then we got G and F, we got F and G again, and then we got E. So what does the glucose get converted into? Identify the compounds labeled D and E. Oh, I'd love to know what they are. So what happens to the glucose? It gets turned into something, and it looks like G and F. Oh, and then we get F and G. This is very confusing because You could get mistaken here easily. Yeah, I think you're absolutely all over it, Matilda. You could probably go for hexose bisphosphate, or you could go for triose phosphate. They should accept either triose phosphate or hexose bisphosphate. It forms hexose bisphosphate first, then breaks down to triose. You might say, isn't triose phosphate E? Doesn't it go hexose, then triose? It does, but look what they've done here. They've reversed F back to G. So they're skipping all of those little steps, and which is what they do. They don't really include all the little steps. It's the overall. So first of all, this is phosphorylation, and then this is the oxidation of triose phosphate into E, which is going to be pyruvate. So E is going to be pyruvate, which happens when you produce the ATP. So G is ATP being broken down into ADP, which is F, and then F is ADP turning back into ATP. Anyway, enough of that. Let's zing down. This is quite a hard question. What is the role of compound D in anaerobic respiration? Jeez. Depends what compound D is. If D is the triose, I've got the answers luckily. I'm going to put them up in a sec. If D is the triose phosphate, then what is the role of D? Well, I wonder. D gets turned into pyruvate, and it's stripped of its phosphate. So we could say the phosphorylation of ADP into ATP. Potentially, so it's helping form the ATP. We could say it's the unstable glucose compound, but we could also say that provide electrons to reduce NAD to NADH. So I'm wondering which of those is going to be the answer for D. So in glycolysis, yes, it could be 2 triose phosphates for the D, two 3-carbon triose phosphates. And then what they do is they provide the phosphate to turn F, which is ADP, back into ATP, which is G. So that's what they get stripped of those phosphates. But they also provide hydrogen and electrons for NAD to become NADH, which we need for the fermentation bit. And then it says, what is, so that's the role of D. Then we've got, why is it important that compound G formed during the reaction in which compound D is converted into compound D? Why is it important that compound G is formed? Oh, hold on. Maybe I'm not looking at this correctly. I don't think I'm looking at this correctly. Oh, I've screwed this up completely. Forget everything I've said in the last 2 minutes. I've just realised something. Okay. Oh. Yeah, so muscle cells. Right, I've just realised, I think that what they're doing here, this is a bit grim. I'm struggling with this question here, guys, which is unusual for me. This is either I'm totally off the park, and then there is another total way this question could go. Is NAD the enzyme in this case? No, NAD is not an enzyme. NAD is a coenzyme. It's an electron carrying molecule. But what I'm worried about is I'm thinking that this is ATP becoming ADP. That's what I was thinking. First half, right? The phosphorylation into hexose bisphosphate or 2-triose phosphate. And then I'm thinking then I'm thinking the ADP in the second half of glycolysis gets converted back. But this says outlines the process of anaerobic respiration. Now, what do we know about anaerobic respiration? So I may have got this bit wrong. Well, luckily I've got the answers here. So it would help if I actually looked at it first, wouldn't it? But you can see how my brain is processing stuff. So maybe that will help. So maybe they're talking about glycolysis. And as it's not specifically said glycolysis, maybe this isn't glycolysis. This is the first bit is the whole of glycolysis, and this is fermentation. And if that's the case, that they're talking, because anaerobic respiration is broken down into 2 parts, if you were here for the start of the live. So it's glycolysis and fermentation. Now, if they're then doing it like that as two overall mechanisms, then that is glucose, that is pyruvate, and G to F is NAD to NADH. So we go NAD to reduced NAD, and then of course the NAD reduced gets turned back into NAD to allow glycolysis to continue, in which case identify compounds D and E. This is pyruvate, and E is going to be, is it in the muscles? It's in the muscle cells. It's going to be lactate. So what my guess is that my first go at this question was wrong, and D is pyruvate. Now, how did I realize that I'd gone wrong? I'll tell you how I realized I'd gone wrong, because I got here and then it said, so then this is lactate. Remember, you could say lactic acid. Why is it not alcohol? Because it's not ethanol, because our muscle cells don't do that. That's what happens in plant cells. Why is the role of compound D, what is the role of compound D in anaerobic respiration? So pyruvate, and then what is it? Why is it important for compound G? And I could be totally wrong. I hope I haven't like reversed on myself. Why is it important that compound G is formed during the reaction in which compound D is converted into compound E in anaerobic respiration? So what am I on about? Why is it important G is formed? Oh, this is to allow glycolysis to continue. So what it is we need glycolysis to be able to continue. So we have to regenerate. NAD. So when the implants ethanol would be E, but this is in a muscle cell, so E would be lactic acid, and we need the formation of E to allow glycolysis to continue. Then compound E is toxic. That tells me that I'm definitely right now. Okay, so definitely right. Rather, I was kind of assuming this was all glycolysis in my first go at this question. Compound E is toxic and is removed from the muscle cell. It is transported to an organ in the body. Which organ is it transports to, and how does it reach the organ? So it's going to be the liver, and it's going to be transported in the blood. So it's transported in the blood. The lactic acid goes to the liver, and there it's made safe by being broken down into carbon dioxide and water. But yeah, that was a bit brutal, that question. I kind of flopped hard on that. So here are your exam board answers for this. Pyruvate, lactate. Oh, I forgot about that bit. What is the role of the NAD? What is the role of compound D, the pyruvate? Hydrogen acceptor. Okay, right. So it takes the hydrogen from the reduced NAD. And then and that is in the fermentation step. So in that step, it does that. So yeah, removes H from NADH, or you could just say removes hydrogen from reduced NAD. This allows glycolysis to continue there, and the liver and in the blood. Anywho, what a disgusting. That was really hard, because I was kind of assuming this was about just glycolysis. So I was like, oh, right, that's the triose phosphate and that's the ATP. But they tend to draw the ATP by convention on the left side of the arrows. And once I got down here and these questions happened, I started to realize, oh, I've cocked this up. Then I reread it and it was specific about anaerobic. So the two stages are glycolysis and fermentation. All right, guys, I'm going to finish there. After that, making a meal of that question. Now I'm going to put a post up for next week, what you want to cover for biology. I'm going to put up some AS topics and an A2 topic, and you can pick, and I'm going to put a post up for chemistry as well. All right, take care, guys. I'm going to finish up there, and I will see you guys again. I'm out.
You're still here?